about the recording. It won't show to you, that's all. It will be recorded automatically. Okay, so now listen every one of you very carefully. Right, in the Telugu Academy book and NCRT, they used to call this chapter as molecular basis of inheritance. Why they have given this definition? Why we are calling it as molecular basis of inheritance? Every one of you listen to me correctly. Molecular basis of inheritance. What is inheritance? Passing of the characters. Passing of the characters from one generation to the another generation in the form of what? In the form of a molecules. That's the reason only they have given the name molecular basis of inheritance. Then why we call molecular biology? Because we are studying the biology of various molecules like DNA, RNA, and protein. That's the reason we call it as molecular biology. Okay, actually friends, this particular slide is the one which will explain the whole lesson. If every student understand this particular slide, everyone will get a clear cut idea what exactly we have to study in the chapter of molecular biology. If you look into the set exam, just from this particular slide, we got 2 questions just from the slide itself. Lot of information I will tell you here itself, and I want everyone to understand. It's a very, very big unit. Now listen, see, you know very well, if I ask you a simple question, what is the function of the DNA? What every one of you give me the answer? Every one of will give the answer that DNA is a hereditary material. DNA passes the information from one generation to the another generation. This is what everyone will give the answer. And if I ask you more specifically, listen everyone, what's the function of DNA? What exactly that, very good. What exactly the DNA is going to do? DNA is going to synthesize proteins. In simple words, every student remember my words. I already told you previously also 37.2 trillion cells present in our body. If they wants to work, the function can be performed with the help of protein only. See friends, without protein, cells cannot function. Let's take an example. Oxygen transport is happening in our body. You tell me, without oxygen transport, any cell can survive in our body? Impossible. Who will transport oxygen? Hemoglobin. What is hemoglobin? Of course, a protein. Now tell me, without enzymes, any biochemical reaction happening in our body, starting from absorption, digestion, metabolism, excretion, everything is enzyme dependent. What are enzymes chemically? Proteins in nature. Suppose example, a antigen enter in our body. What our body starts synthesizing? Our body starts synthesizing antibody. What is antibody? Protein. So what I mean to say, every function performed by the living cell is under the control of protein only. That particular protein can be synthesized by a versatile molecule, the DNA. That's the reason, friends, we can call DNA is the most abundant molecule in our cell. Okay, now if the question asked which is more important, DNA or protein, undoubtedly friends, DNA only. Why? Because the protein can be synthesized from the DNA only. Without information from the DNA, you can't synthesize the protein. It is impossible. Now here comes a wonderful question. I just said to all of you that DNA is the one which is going to synthesize the protein. There is no doubt about it. Now the question is crystal clear. If the DNA can synthesize the protein, why not DNA can directly synthesize the protein? Why DNA is going to synthesize an intermediate molecule called RNA? My question is very clear. I only said that DNA can synthesize the proteins. Then why the DNA is not synthesizing the protein directly? Why the DNA is synthesizing an intermediate molecule called RNA? Everyone, can anyone quickly tell me? Wonderful. Because DNA is a very highly stable molecule, extremely stable. So what you have to understand, friends, in order to protect its stability, in order to maintain the integrity, in order to protect from the errors or the damages, DNA is not directly involving in the synthesis of protein. Instead, DNA is going to synthesize an intermediate molecule called as RNA. Now you can ask me, sir, what happens if the RNA get damaged? There is no problem. If the RNA get damaged, we have DNA, right? DNA can synthesize RNA. RNA can synthesize the protein. So, but if DNA get damaged, the same genetic information passes to the next generation. Obviously, it leads to the lot of chromosomal abnormalities, lot of genetic defects. That's the reason DNA never involved in the synthesis of protein directly. Now, every student try to understand my words correctly. The DNA, before synthesizing RNA, DNA undergoes what process, friends? DNA undergoes a process called replication, which we also called as duplication, which we also called as cloning. We are all looking like our mother and father, and your son and daughter look like you and I mean to say, we are all looking like our parents. Our next generation looks like us. So what is happening over there? Our genetic material is passing from one generation to the another generation in the form of duplication or the cloning or the replication. Now, once the DNA undergo the process of replication, what did I told you, friends? DNA is going to give rise to the RNA. What most of the students will think that whenever DNA is going to give rise to the RNA, students will think that the entire DNA is going to convert it into RNA. That is wrong statement. See, only a small part of the DNA. See here, I'm writing only a small part of DNA getting converted into RNA. Now you have to ask me a very good question. Sir, DNA is a very large molecule. Which part of the DNA is getting converted into RNA? That's a very good question. Because it's a long molecule. So which part of the DNA can convert into RNA? See, friends, it dependent on which protein DNA has to be synthesized. Try to understand the concept correctly. Suppose example, this DNA coding for 1 lakh proteins, example. So suppose I want to synthesize hemoglobin. Okay. Now the DNA contain information for hemoglobin. Only that hemoglobin part of the information of the DNA can be converted into RNA. Then the RNA can synthesize that particular hemoglobin. Very simple point. So you people should not think that the entire DNA is going to be converted into RNA. Never, ever. Only a little part of the DNA, a small piece of the DNA can be converted into RNA. So friends, remember my words correctly. DNA mediated RNA synthesis. What is the definition I'm giving you? DNA mediated RNA synthesis, DNA guided RNA synthesis, called as transcription. No need to get confused with the point. Okay, now you can ask me, sir, what the RNA brought? You only understand right now, RNA brought the information, is it? It brought abundant amount of the information. So it brought the abundant amount of the information from the DNA. That information, with the help of amino acids, it is going to converted into the protein. So my friends, what is the definition I'm going to give you? RNA guided, RNA mediated protein synthesis is called as translation. But you have to ask me, sir, who is controlling the protein synthesis? I can tell you, like DNA only. Okay, so you have to understand, you can consider DNA is like a father and RNA is like a mother. Father is the one who will control the entire family. So imagine the family is like a protein. Okay, but mostly a father will control the entire family with the help of a mother. Is it? Same way here also, DNA is the one which will do everything. But DNA won't directly involved in the synthesis of protein. Instead, it using the RNA as the intermediate molecule. Do not get confused with the point. Now, friends, listen. Where most of the students will make the mistake when they ask the enzymes. 90% of the questions will come from the enzymes. Hence, it is utmost important. Every student has to remember enzyme. So listen this slide very carefully now. So DNA is undergoing the process of replication. Of course, true. So if the DNA wants to undergo the replication, there is a requirement of enzyme. What is that enzyme called as DNA polymerase enzyme? Now the first question comes in the exam. Remember, don't confuse. DNA replication means what? Every student look into the picture. I am synthesizing the DNA from the DNA only. Correct na? So the enzyme involved is DNA polymerase. Now the question is here, from where the DNA polymerase is getting the signal? Who is controlling the DNA polymerase enzyme? DNA polymerase enzyme is controlled by the DNA only, friends. That's why we can call it DNA dependent DNA polymerase. Every student will confuse. Remember, DNA dependent DNA polymerase. Okay, that's fine. The replication is over. Now, DNA is going to give rise to what? RNA. Who will synthesize the RNA? Do you think DNA polymerase will synthesize the RNA? Never ever. DNA polymerase only synthesizes the DNA. What does the RNA polymerase will synthesize? RNA polymerase will synthesize the RNA. But that is not the question. What is the question? Who is controlling the RNA polymerase? Who will control? RNA polymerase cannot controlled by RNA. You should not think that. Since we have not synthesized RNA only, how can RNA polymerase can be controlled by the RNA? That is not the correct answer. DNA is controlling the RNA polymerase. So listen my statements, everyone. DNA dependent RNA polymerase. It is not RNA dependent RNA polymerase. So what is the function of RNA polymerase? Synthesizing the RNA. From where? From the DNA. Now here comes a very important exception. You all people know very well, every organism having DNA as their genetic material, but we all know very well, most of the viruses are having what? RNA as their genetic material, not the DNA. So now every student listen. Now I have to synthesize DNA. From where? So I am see what I am drawing now. I am going to synthesize DNA from the RNA. So every student has to remember the point. When I use a word, I want to synthesize the DNA. Every student has to remember a statement that I want an enzyme called as DNA polymerase enzyme. It is the fixed point. If I want to synthesize RNA, I require RNA polymerase. But here the question is very clear. I am synthesizing the DNA from where? From where I am synthesizing? I am synthesizing from RNA, which means RNA is guiding me. So what I will be called as? I will be called as RNA dependent DNA polymerase enzyme. Widely called as what enzyme? Reverse transcriptase enzyme. This is the question asked in your previous JL and DL also, as well as in the set exam also. And you all people commonly know that. So listen, every student, DNA dependent RNA polymerase enzyme will be involved in the synthesis of RNA. Similarly, RNA mediated DNA polymerase, which is also called reverse transcriptase, which synthesize the DNA from the RNA. Please don't get confused with this particular point. And friends, once we synthesize the intermediate molecule RNA, what is going to happen? Now the RNA is going to synthesize what, friends? Protein. So RNA mediated protein synthesis called as translation. It's a very important point. So now you use your brain, Right now, from the RNA, we found a mechanism that we can synthesize the DNA. Till today, the research is happening whole world. Is there any mechanism is present in our cell that we can synthesize RNA from the protein, that we can synthesize even DNA from the protein? Still the research is in progress, but we really don't know that kind of mechanism is really present in the cell or not. But still the research is in progress. So you understand the enzymes. Now the most important point for the exam is where all these are all happening. That is very, very important, friends. Where these processes are happening in the prokaryotic organism also and eukaryotic organism also. Because without location, you never understand the concept very correctly. In case of a prokaryotic organism, where the replication will take place in the cytoplasm, because you know very well bacteria do not have anything except cytoplasm. You know that. So in case of eukaryotes, we are well developed organism. We are highly advanced organisms. So in the eukaryotes, where it will happen in the nucleus, because we are having a well developed nucleus. And if you look into the transcription, where the transcription happen in the prokaryotes, once again in the cytoplasm, where the transcription happen in the eukaryotes nucleus only. But my friends, let me tell you one important point to all of you. Whether it is a prokaryotic organism or whether it is a eukaryotic organism doesn't matter. The protein synthesis always takes place in the cytoplasm only. What is the reason? Because in prokaryotes and eukaryotes, if the proteins want to be synthesized, we require a lot of factors. We require a lot of enzymes. All those enzymes and factors are present in the cytoplasm only. That's the reason protein synthesis will take place only in the cytoplasm, whether it is a prokaryotes or eukaryotes. Now I'm going to tell you the most important point everyone has to remember. This is also most people will get confused. Imagine this is a prokaryotic cell, okay? And imagine this is a eukaryotic cell. Everyone pay attention. See friends, just now I told you very clearly, in the prokaryotic cell, what is happening? DNA replication is happening in the, remember everyone, in the cytoplasm. All the students understand, whenever the DNA replication is over, from the DNA which is coming out, every student look into this, RNA is coming out. Correct na? Now all the students remember, whenever RNA coming out, listen my words, if 20 nucleotides of RNA comes out enough, immediately, you know friends, what is going to happen? Immediately ribosome will come and attaches there. Immediately ribosome will come and attaches and immediately start the synthesis of protein. Now friends, what is the statement I want to conclude? I want to conclude a statement that in case of a prokaryotes, transcription and translation both are coupled. What do you mean by the word coupled here? Both are happening simultaneously. Try to understand, do not get confused. The moment mRNA comes out in the prokaryotes, immediately ribosome assemble and ribosome immediately start the synthesis of protein. Never ever confuse with that point. But friends, do you think it happens in the eukaryotes? No, right? Because we are having well developed nucleus. So what's happening here? First, DNA replication happen in the nucleus. From the DNA which will synthesize, mRNA will synthesize. But friends, the interesting point here, whatever the mRNA that is synthesized in the eukaryotes, it is not active mRNA. Remember my statements, what I'm using the word, it is not biologically active. So what I called that mRNA, I can call immature mRNA. You all people studied, right? If I want to convert the immature mRNA into mature mRNA, what I have to do? I have to do the modification. That is also called as RNA processing. We will study elaboratively. What are the various steps in the RNA processing? We will do 5' capping. We will do 3' polyadenylation. We will do splicing. You all people know it very well. Now, question is very clear. When these modifications are happening, that is the question. When these modifications are happening, these modifications will happen after the transcription. Yes or no? Since they are happening after the transcription, we called them as post-transcriptional processes. What do we call, friends? Post-transcriptional processes happening after the transcription. Without post-transcriptional processes, mRNA in the eukaryotes never ever become matured. But that is also not the question. The question is that where these post-transcriptional processes are happening, they are also happening in the nucleus only. That's an important point, friends. So now the next and very important point. Okay, I have synthesis. Then after the modification, mRNA from the nucleus, it enter into the cytoplasm. Then it will synthesize the protein. All the students imagine my words. In the prokaryotes and in the eukaryotes, the protein synthesis is happening in the cytoplasm only. Now you tell me, once the protein got synthesized, can you call it as a active protein? No, you never called it as active protein. You can call it, see, all the students listen. Most of the people will get confused between naive protein and native protein. All the students remember, naive means inactive protein. Native means biologically active protein. Once again, you have to understand, if in order to convert the naive protein into native protein, what kind of modifications we have to do? We have to do post-translational modifications. Why did I use the word post-translational? Because they are happening after the translation. So now I am going to summarize everything. In case of a prokaryotic organism, all the students listen. Replication, transcription, translation, and post-translation. Everything will happen in the cytoplasm. Every event will happen in the cytoplasm. Which is not happening in the prokaryotes, tell me? Post-transcriptional processes never ever happen in the cytoplasm. Why? What is the reason? Because transcription and translation are happening at a time in the prokaryotes. But friends, if you look into the eukaryotes, what every student has to remember: replication, transcription, post-transcriptional processes, even RNA editing also, everything happen in the nucleus. What are those? Replication, transcription, post-transcriptional processes, and RNA editing. They will happen in the nucleus. When it comes to the translation and post-translation, they will happen in the cytoplasm. They can ask a very direct question: which of the following steps will occur in the cytoplasm in case of a eukaryotic organism? Only translation and post-translation happen in the cytoplasm, remaining everything in the nucleus. In case of prokaryotes, everything in the cytoplasm only. There is a no event called as a post-transcriptional processes in the prokaryotes. Such a simple point it is. So you have to remember all the events compulsory, all the enzymes compulsory. So this is what molecular biology is. So now what we have to study in molecular biology? We have to study the replication in prokaryotes and eukaryotes. We have to study transcription in prokaryotes and eukaryotes. We have to study translation in prokaryotes and eukaryotes. Last step we can call gene regulation in prokaryotes and eukaryotes, and DNA repair. This is the syllabus of molecular biology for the set or net or JL or DL. Everything is same, but the level they ask the questions are different. Okay, so now first step is what, friends? Replication. You only look into the picture. Information is passing from where to where? Information is passing from the DNA to the protein. So this passage of information from the DNA to the protein level, we can call central dogma, which is proposed by a genius molecular biologist, Francis Crick. What the Crick said? Crick said that the process by which information is moving from the DNA till the protein level, that is called central dogma. Don't get confused with that point. So now, what is our first step in the central dogma? That is the synthesis of DNA. Don't think that the synthesis of DNA is that much easy process. Very complicated process. Compared to DNA synthesis, RNA synthesis is more difficult. When compared to RNA synthesis, protein synthesis is more difficult. So this chapter is like this. If you do not understand one step, you never understand other steps, because all are interlinked like immunology. Okay. Now listen, everyone. So now we are going to start first thing, replication. When I said replication, you can't study the process directly. What you have to study first you have to study rules of a DNA replication. Next, you have to study enzymes of a DNA replication. Then you have to study the process. See, if you want to make a curry, you can't directly prepare the curry. First, you have to cut the vegetable, you have to boil the oil. Same. You can't do the process directly. First, you have to follow some rules. You have to prepare all the enzymes. Then only you can do the process. So very complicated process it is. So now let's look into this. What's happening here? You all people know that the two genius molecular biologists, James Watson and Francis Crick, who proposed what structure? Double helical structure of the DNA. Every one of you remember my words. I did not keep the photo. Before these two people, the teacher of these two, his name is Paul. You know, that time he's also very leading scientist. He's also molecular biologist, the teacher of these two people. Paul, he proposed, friends, triple helical structure of the DNA, not the double helical. He proposed the triple helical structure of the DNA, but unfortunately, most of the other scientists never accepted this model because there is no evidence also. There is no proof to prove that the DNA exhibit in the triple helical structure. So that's why this model was failed. Later in the year 1953, these two people identified that DNA can exist in the double helical structure. Of course, the question they can ask in the exam, what question they can ask you in the exam, which technique is used to understand the double helical structure of the DNA? That's a direct question. So by the X-ray crystallographic structure, I want everyone has to remember. And who has taken the X-ray crystallographic structure of the DNA? It is taken by the Rosalind Wilkins. Rosalind Wilkins and James Franklin. Okay. In the molecular biology, genetics, and ecology, everyone has to remember the name of the scientist. It's very important, friends. Now the question is here, first Nobel Prize was taken by the Watson and Crick, but later Nobel Prize was shared among the Wilkins and the James Franklin as well. Okay, but before receiving the Nobel Prize, only Wilkins passed away, but on her memory the later it was shared. Okay, so there are total 4 scientists are there. These two identified the structure based on the X-ray crystallography taken by the Rosalind Wilkins and James Franklin. Okay, now listen to me correctly. Now we are going to study the first step of our central dogma, that is the DNA replication. What did I told you very clearly? DNA see, the more crystal clearly you will understand the concept, it becomes very easy. If you get a confusion, ask me immediately. Now, what is replication? Now, if I ask you a question, please define the DNA replication. Give me a biological definition and give me a chemical definition. What you can define me? If I ask you to please give the biological definition, what you can say? DNA replication means there is a cloning of the DNA. How does the cloning occur? All the students try to understand. Whenever the duplication is happening, whenever the cloning is happening, look into the picture. The two parent strands are getting separated. You can see here, this is one parent strand, this is another parent strand. My friends, when the parent strands are separated, they are acting like a templates. What? Template means which will help the other strand to get synthesized. They will act like a template. Once they are acting as a template, you can see here, there is a synthesis of the daughter strand. So now, how can I give the biological definition? I can give the biological definition. DNA replication is a process in which what are synthesizing? Daughter DNAs are synthesizing by using parental DNA as the template. That's a simple definition I will tell. So now you can ask me, what is the ultimate goal of the DNA replication? Ultimate goal is what? We have to transfer the genetic information to the descendant generation. Descendant means next generation. See friends, the most unique point everyone has to remember, you know why the DNA replication is very popular, famous when compared to the transcription and translation? Because of fidelity, accuracy. See, you know very well DNA consists of billions of bases. You know how long DNA molecule? You take our DNA, keep it on the floor. From planet Earth, it will go to planet Mars. Once again, it will come back from planet Mars to the planet Earth 600 times. That much long molecule the DNA is. So made up of billions and billions of nucleotides. So doing the accurate replication of billions of DNA nucleotides is not a simple task. But the versatile DNA polymerase enzyme will do it very accurately. That's why we can say that DNA replication is a very high accurate process when compared to the transcription and translation. So this is the simplest biological definition. And most of the people will ask one simple doubt. Sir, what do you mean by the word synthesizing the daughter strand? That's a very good, valid question. These two are parent strands. What do you mean by the word synthesizing? Synthesizing means adding. What I am adding? I will add one nucleotide with another nucleotide. By that, I can synthesize the daughter strand. So friends, what is the chemical definition I can give you? Adding of one nucleotide with the another nucleotide. That is called DNA strand synthesis. See, I will explain in the next slide. You know, right? DNA having a deoxyribonucleotides. See, all the students remember my words. When one nucleotide is going to add with the another nucleotide, always will add in the form of a 3 phosphates. Every student has to remember why 3 phosphate. I will justify it in the upcoming slides. DNMP means deoxyribonucleotide monophosphate. N represents number. DNTP means deoxyribonucleotide triphosphate. So 3 phosphates are coming. Out of 3 phosphate, only 1 phosphate will be added. How many phosphates are left out? 2 are left out, released out. So it become N plus 1. Later another DNTP will come. It will become N plus 2. Another DNTP will come, N plus 3. Like that, billions and billions of nucleotides will be added. So now what is this DNMP and what is this DNTP? Very, very important. You have to remember them. Nucleotide is made up of 3 components. Everyone remember, nucleotide is made up of a sugar. Everyone has to answer. A phosphate group, a nitrogenous base. 3 together, you all read it, call it as nucleotide. And look into the point, if I remove the phosphate group, then what is left out? Sugar is left out, phosphate is left out, sorry, sugar is left out or base is left out. I can call it nucleoside. See, now every student answer my question. Now let me talk 2 things very correctly, individually. Let's talk bases individually. Let's talk sugar individually. This is how many carbon? See friends, whether it is a DNA or whether it is the RNA, sugar is called pentose. You no need to remember the structure. I want you to understand the structure. Pentose means how many carbon? 5 carbon. So the pentose of 2 types. One is called ribose sugar. Another one is called deoxyribose sugar. Why I am calling it as the ribose? Why I am calling it as the deoxyribose? All the students look into this. First carbon, second carbon, third carbon, fourth carbon, fifth carbon. Then why I am calling ribose and deoxyribose? It is depending on the second position. My friends, listen. If the second carbon, if it contain OH group, if the second carbon, if it contain OH group, we will be called as ribose. Instead of the OH group, if the second carbon only contain hydrogen, you only see the definition deoxyribose. D means no oxygen. If there is a no oxygen is present, if only hydrogen is present, we can call it deoxyribose. So the question is very clear. In our DNA, deoxyribose is present. That's why we can call deoxyribonucleic acid. Based on the sugar only, we have given the name. But if you look into the RNA, there is a presence of OH. That's why RNA called as ribonucleic acid. Never get confused. That's why we have given the name. So now friends, how many carbons are there? 1, 2, 3, 4, 5 carbons. But friends, everyone listen, look into the structure correctly. Only 4 carbons are present in the ring. Correct na? Where is the 5th carbon? 5th carbon is out of the ring. You can call it arm position. So friends, how many carbons are there in the ring? 1, 2, 3, 4 carbons with 1 oxygen. Everyone please answer the question. 4 carbons plus 1 oxygen. What do we call this ring structure? Everyone, what is the name of the ring which consists of a 4 carbon plus 1 oxygen? Not even 1%. You have studied in the +1 level. Sangeeta said pyrrole. That is absolutely wrong, Sangeeta. Very good, very good. See, it is called a furon ring. Every student remember, don't confuse. When 4 carbons plus 1 oxygen is present, it is called furon. Since it is a sugar, right? We add the OSE, it becomes furanose. So pentose is a furanose. Now every student answer. Now let's take a glucose. Glucose having how many carbons? You know, right? 6 carbon. But in the ring, glucose having 5 carbons plus 1 oxygen in the ring. What do you call this structure, everyone? Everyone, what do we call this structure? This is called a pyron ring. So it is a sugar, we can call it pyranose. Don't confuse. Okay, it is called what? Pyranose. So don't confuse. Pentose ring is called as a furanose ring, and glucose ring is called as, or hexose ring is called as pyranose. Never forget. So this is the sugar. And what did I already told you? Phosphate, alpha, beta, gamma, mono or mono, di or tri. My friends, we always used to say that DNA is a negatively charged, because friends, the charge is very important. We have studied, please do not disturb me, don't turn on videos and audios. I have told to all of you very clearly that DNA is negatively charged. You remember, if the DNA wants to be converted into heterochromatin or euchromatin, every student remember, it is depending on the phosphate group only, friends. So that phosphate is the charged one. DNA is charged because of phosphate group. Never ever confuse with that point. Now look into the picture. There are two bases are present, which is called nitrogenous bases. Now you try to understand how important nitrogen in our body. Without nitrogen, we can't do anything. Without nitrogen, you can't synthesize the DNA only. That much important it is. So it consists of adenine. So two bases are there, purines and pyrimidines. Sometimes in the exam, they will give the rings like this. Purines means bicyclic, means two rings are there, right? So they indicate like this. Pyrimidines, they will indicate like this. Only one ring is present. Never ever confused with that point. According to the Chargaff's rule, we're going to study it. Purines are always equal to the pyrimidine, which means, please mute your mics. If purines are present 50%, pyrimidines are also present 50% in the DNA, which means that number of purines are always equal to the number of pyrimidines. What are the various purines, friends? Adenine, guanine are said to be the purines. Cytosine, thymine are said to be the pyrimidines. And you know very well, RNA also contains same. But in case of RNA, thymine won't be present. It is replaced by the uracil. Now, I told you many times last time also in the DNA repair. Sir, uracil is present in the DNA or not? Friends, because of many mutations, uracil is very commonly found in our DNA. But of course, our repair enzymes will remove it. But you remember, uracil is commonly found in the DNA also because of the mutations. Now, adenine always pair with thymine with 2 hydrogen bonds. Guanine always pair with the cytosine with 3 hydrogen bonds. That's a common point. So now you know what is nucleotide. So this is called one nucleotide, correct? Now that is not important, reading what is nucleotide. What is important for exam? You have to understand the bonds. How? See, I have explained you sugar differently. I have explained you phosphate group differently. I have explained you bases differently. Now I want you to know, how can I join these three? That's the question. See, remember everyone, base and phosphate are connected via sugar. So sugar is doing everything. Sugar is important now. So now the first carbon, what carbon it is? First carbon. First carbon of the sugar can be connected to the nitrogenous base. This is what do we exactly call, friends? We exactly call it as N-glycosidic bond. My friends, wherever you heard the name of a glycosidic bond, remember it is the sugar. Sugar connecting the base. I already told you now, most of the students will think that, see, this is the first carbon connecting the base called glycosidic bond. This is which carbon, friends? Fifth carbon. Fifth carbon connecting what group? Phosphate group. Every student will say that it is the phosphodiester bond. Everyone keep this point in your mind. You never call this as the phosphodiester. You call it as ester bond. Okay. You can also call widely you can call anhydride bond. This is not a phosphodiester. I want you to remember the point very correctly. It cannot be phosphodiester. Now see, let me explain you what is phosphodiester. Okay. This is one nucleotide, right? Now, friends, one nucleotide is going to join with another nucleotide. How is that possible? Can we join one nucleotide with another nucleotide? Of course, we can join. Every student look into this. This is the sugar. And to the sugar, what is added? Base is added. Correct? Now, everyone has to understand this. This is very important. And at the fifth carbon, phosphates are attached. This is one nucleotide. So let me draw the another nucleotide. This is another nucleotide, having the sugar, having the nitrogenous base, having the phosphate group. Correct? Na? So there are two nucleotides I have drawn. Every student look into the diagram very correctly. You only look into the picture. Since we are talking about DNA, right? You tell me, friends, do you think DNA having OH group in the second carbon? No. But every student look into the picture. DNA at the third carbon, the sugar contains OH group in the third carbon. Now OH group is present here. Correct or not? Now every student understand you. OH means what? Oxygen is present, hydrogen is present. Oxygen act as a nucleophile. You all read in the chemistry. Oxygen can act as what? Nucleophile. What are these two dots? They are the lone pair of electrons. What are those? They are the lone pair of electrons. What that lone pair of electrons will do? Listen everyone. It will come and cut one phosphate. When it will come and cut one phosphate, how many phosphates are there? Two are there. So those two are released in the form of PPI. This is what energy. Now, where the bond is formed, you only look into this. The bond formed between 3' hydroxyl group of one nucleotide with the 5' phosphate of the another nucleotide. This bond is called phosphodiester, which we can call backbone. What bone? Backbone of the DNA. So always backbone means which is formed between the sugar and phosphate. Now tell me, like this third nucleotide is added. So second nucleotide having OH group here, and once again it will come and join with the phosphate groups of the last nucleotide. So friends, now listen every one of you very correctly. Just imagine friends, just imagine this is the first nucleotide, right? First nucleotide it is. You tell me, first nucleotide contain phosphate group. At which carbon phosphate groups are present? Fifth carbon phosphate groups are present. Correct na? Now tell me, this phosphate groups reacted with any other group? No, not at all. It is not reacted with any group. That's why it is called 5' direction. Now imagine friends, this is the last nucleotide. The last nucleotide contain OH group. Do you think this OH group can react with any other phosphate group? Never ever, because it is only last. The OH group is present at which carbon? Third carbon. That's why it is called 3'. So now you understand why we call DNA as one strand as the 5' to 3'. Other strand is opposite, right? Then you can call 3' to 5', opposite strand. That's what we can call the 5' to 3', 3' to 5' based on their sugar and phosphate arrangement. Never ever get confused. Whatever I am explaining is so simple basics, because you have to know it, and don't think that you will get the question from phosphodiester bond and anhydride bond. You never get that questions. Okay, you never get that questions in intermediate also, but you have to know that in order to understand the concept. Okay, so now as I told you, if you want to read the replication, first you have to understand the rules. And most importantly, you have to understand the enzymes. After understanding rules and enzymes, this will become super easy. Now let's focus on the various rules. Now the question is very clear. Do the DNA has to really follow these rules in order to undergo the process of DNA replication? Of course, yes. You all people are applied for JL and DL. If you are not meeting the eligibility criteria given by the government, of course you are not eligible to become JL and DL. Same way, if the DNA is not following these rules, DNA never undergo the replication. The replication will immediately stops by the enzymes. So what are those rules? There are many rules are there. One is called semi-conservative. We will explain one by one. Starts at the origin. Very, very, very important rule. The synthesis should always occur in 5' to 3'. It can be uni or bidirectional, semi-discontinuous. RNA primers are required, high fidelity. And the interesting point is that, see, 3 plus 4, 7 rules are there. If DNA miss even one rule also, the replication never happens. If 7 rules are there, it has to follow the 7 rules compulsory. Every rule, every stage, the DNA has to qualify in order to undergo the process of duplication. So now let's look into the one by one. One is called semi-conservative model. It's not a difficult model, but it is a confusing model. Here you no need to remember even a one point also, everything to be understood. First of all, you have to ask me, sir, what is this model? See friends, if you want to understand anything when you are preparing for competitive, remember this: what, why, where, when, how. If you apply these 5 points, how difficult the concept it may be, it will become easier. Never try to by heart, try to apply these terms. Now listen, what is this semi-conservative model? We studied that our Watson and Crick proposed the double helical structure of the DNA. And Watson and Crick only said that DNA undergo the process of replication. Use your brain. If the DNA wants to undergo the replication, does it undergo the replication randomly? No, right? Suppose you are in a Tirupati queue. Suppose you are going to a temple. Okay. So if you are going to a temple or if you are in a church, you will always move in the queue. Correct, right? So you never move randomly. Correct, na? So you can't directly go. You have to move in a particular queue. Same, DNA wants to undergo the replication. DNA should not undergo the replication randomly. It has to follow a model. When it follow a model, then it undergo the replication very correctly. What is that model? That model only called semi-conservative model. What is that model? How can I define that model? According to the semi-conservative model, during the process of DNA replication, the listen everyone, the two parent strands of the DNA can get separated, and each parent strand acting as what? Template. And it is going to synthesize what strand? It is going to synthesize the daughter strand. Correct, na? So this is what do we call semi-conservative model. Who proposed this model? So Watson and Crick said that yes, DNA exactly undergo the replication like this only, where two strands are separated and daughter strands will be synthesized. They said this. But what is the proof? See, we know that Watson and Crick only proposed the double helical structure of the DNA. So, but they are only saying that DNA can undergo the replication by using this model. But where is the proof? So there is no proof. There is no evidence to prove this. So some scientists accepted this model. Some scientists opposed this model because there is no proof. So some scientists opposed the model and they proposed their own models. So this is semi-conservative proposal by Watson and Crick. See this? This is called conservative. What is the conservative model? Look into the picture. Don't confuse. This is the entire parent DNA. You can see, right? Entire parent DNA can be preserved. Listen my words. Entire parent DNA preserved or conserved. Conserved means preserved. And it is giving rise to daughter DNA. What I'm saying? Entire parent DNA conserved or preserved and giving rise to the daughter DNA. That's why it is called conservative model. Friends, then some scientists propose another model called dispersive. When I use the word dispersive, remember cut and patch. Cut and patch model. It's like a small parent, small daughter. Look into this. Blue color is the parent, yellow color is the daughter. Parent, daughter, parent, daughter, parent, daughter, parent, daughter. This is called cut and patch. But here comes the question. What was the question? Watson and Crick proposed semi-conservative. Some other scientists proposed conservative and dispersive. But we really don't know. Our DNA molecule will follow which model in order to undergo the process of replication. We really don't know. So to prove this, two genius molecular biologists named as Matthew Messelson and Franklin Stahl, they have conducted a beautiful experiment. Finally, they have proved, yes, DNA always follow semi-conservative model only while undergoing the replication. DNA never follow conservative, DNA never follow dispersive. It only follow this model. Till today, we can consider this is one of the most beautiful experiment that happened in the field of biology. Okay, how it will happen, we will study in the tomorrow class. Okay, so I want you to revise immediately, okay, on the same day itself. So by today evening only, you will receive all the notes for the mol bio, biochemistry, genetics, plant physiology, ecology, cell biology, and unit 1, 2. I will send all the PDFs separately, chapter wise, but never neglect the PPT. Okay. Apart from PPT and notes, first listen my lesson. Okay. So if you have a good idea, then it's easy to read. Okay. So please read. By Sunday, you people has to write the exam till whatever I complete on the Saturday. So mostly I'll cover a lot of concepts by Saturday. You have to write the exam. Okay. Is it clear, everyone, so far? Now, if you have any doubts, you can ask me. Any doubts? Please ask any doubts if you have. Clear, everyone? And I want everyone to attend 5 p.m. without fail. Okay. I will start the RDNA technology. Okay. Please, very fast we have to go. Perfect then. Okay. I want you people to revise it thoroughly. I'll share the PPTs as soon as possible. Okay. Thank you all. Thank you. I'll see you in the evening. Thank you.